Yep.
Yeah,
Yep.
thanks very much. Thank you. Okay, so it's just a basic cellular unit of the nervous system. Each neuron is composed of a cell body, normally referred to as the soma. It's fine branching extensions, dendrites, which take the information on initially. They receive incoming nerve signals and a signal long extension, which is an axon. It's a slender projection. Again, I spoke to you a short bit about this maybe in the first evening, and it conducts nerve impulses. So really what the APA definition doesn't have in there, and I've mentioned this to you before, you can never really go wrong with a neuron when you mention that it's an electrical chemical messenger. Okay, it's really an impulse. They call them spikes sometimes because it's uh, electrical chemical impulse. Okay, electrical chemical messenger, whatever way you want to refer to it. That's what we speak of when we speak of the neuron then again. And you will get this set of notes then again sent to you after. This is what a general motor neuron will look like. So we've different types of neurons. Again, we will get to that in a short while. There's 10 real parts to it that I often say to students, not that you will need to know it in any great detail, but for people who are interested in the psychology end and will go on to study maybe an undergraduate degree in this is something that you will probably come across then again. The whole thing together is called an action potential, and anybody who studied biology, particularly at honours level in the Leaving Cert, will be fully aware of this. This is the messenger that we use to do everything that we are in terms of our thought process. One fires away, connected at the dendrite, and then it will pass on at the other end then at the axon terminals. All of the parts in between, I'll mention a couple of them here and there, but they're not too dissimilar. So these are the neurons and the neural pathways that go on in between everybody's brain. So there's a nice picture of me with the neural pathways that are there. You can see how diverse that they are. 100 billion neurons we have housed within our brain, within our body, should I say. About 86 billion of them are all within our brain then again. So if we are anything in terms of function, neurons are vitally important to us. Okay, so you think back, you think chemical messenger, that's what it's doing. They're sending messages from one to the next to allow us understand that. Again, what is biopsych? The science which attempts to explain behavior in terms of biology. Now, it's not a great thing to say, but really the biology of the brain and the understanding of it often is better understood when something goes wrong. So when we think of Parkinson's disease, Alzheimer's, any of these things, dementia, any of these things that occur, that's when we start to really understand localized function of the brain. Okay, and there's a really big figure there. That them images probably don't mean anything at the moment, but they will when we speak about Phineas Gage. Phineas Gage, railroad worker. You may have heard of him in the past. On the left there's a museum. I'm not sure to why that's there, but this iron rod shot up through at the angle that's there, up through his cheek then and behind his eye. Okay, the thing with Phineas Gage, why he is so famous within psychology, is that we learned so much about biopsychology off Phineas Gage, and really because there's a famous phrase within psychology that after a couple of weeks, like he was conscious while all of that happened. By the way, he remained conscious, went with his friends or colleagues to a doctor, to a physician. The physician said, there is no way that this has happened like you're telling me. He began to cough, as the story tells us, and bits of brain started to come out. But consequently, what happened was his personality changed, and the phrase within psychology is that he was no longer Gage. So the personality type changed, and his own understanding of his day-to-day existence changed because of the part of the brain that was well, I wouldn't say destroyed, but was definitely damaged and impaired from what happened then. So you can look it up. There's loads of material on it. There's loads of confounding stuff in terms of whether it's true and little bits and bobs. But what we do know is that there was journals in around the time. In 1848, there even a passage of an iron rod through the head. Harlow then said recovery from the passage of an iron bar through the head. These things were all physicians around the time who looked at it. So it's definitely a true instance. It's definitely a true event, and it just led into giving us an understanding of, hang on one moment here, we are aware, although looking at the time 1848 in around them, that the biology of the brain is a lot to do with what and who we are. Okay, so again, that just explains what we looked at the last time there. And really we look at the central nervous system. So again, I think I mentioned this for people who've got an understanding of anatomy and physiology. Fantastic. If not, do not worry. Your central nervous system, generally classified as your brain and your spinal cord, as the image shows there. In some of the literature, it will be classified as three. And I'll never forget, we got this question wrong, me and a couple of people who we were at a quiz in the US, and it's classified as three parts within the US: the brain stem, and the spinal cord. The brain stem, tiny little part at the back of the brain, generally classified as part of the brain within a lot of the European, particularly the Eastern European literature. But you will find in some of the literature there that it's titled as three parts then. So this is the way it works. You'll often hear people talking about the nervous system and how it works. So really, the central nervous system is what I said. So the central nervous system is the brain and the spinal cord, okay? Then what is the peripheral nervous system? So if I was to say to you now, oh, look, this player was on the periphery of a game, you'd be thinking that they weren't really involved. They were on the outside of the game, weren't really. And really what it's telling us here is it brings information into the central nervous system and it carries signals out of the central nervous system. Me doing jazz hands in front of the screen like this is part of the peripheral nervous system. That message was initially sent from my brain down the spinal cord and out across this central nervous system into the peripheral nervous system that allowed me to perform jazz hands in front of the group. So on the left hand side, you can see it's pretty much broken down into the physiology of it, the brain and the spinal cord as such. And then on the right hand side, the peripheral nervous system is often intimated as the somatic nervous system and the autonomic nervous system. Now, the autonomic nervous system, as you will see here now, it refers to the involuntary unconscious functions, okay, and specific ones that are associated with it then again. So our somatic nervous system will often be talking about these voluntary movements that we have. The sympathetic division of the autonomic nervous system is fight or flight, or freeze, as we've said before, but freeze generally doesn't take on any of them sympathetic observance. The parasympathetic division then is rest and digest. That's the things that when we do rest, when we are at peace, we can just relax, I guess. It's not like I eat my dinner and then I sit there and I say, okay, stomach, esophagus, get ready as we're in action now, boom, boom, boom. It's not like that. These are all done, as we've said before, automatically. So they're involuntary functions as such. So them involuntary functions happen. Now you think about what happens when we eat late at night. What does it do to our sleep patterns? Well, this rest and digest, it doesn't really get to do that because your digestive system, beginning with your esophagus and obviously ending with your sphincter, then thinks, well, I've got the night off. But actually, if you've eaten close to the time that you're going to sleep, your digestive system is still switched on. So not only is it the chemical part of your digestive system, it's the mechanical part of your digestive system. So the mechanical part is just your teeth. It's chewing the food, it goes down, peristalsis happens, it pushes the food down, and then the chemical process takes over. But the chemical process cannot switch off either because obviously it's digestive enzymes. If you think of something like your appendix, we would have eaten, highly cellulose food that would have been highly difficult to break down. And essentially that's why we had the appendix. It's not really used the way it would have in the past, okay? Your functional divisions then of your peripheral nervous system, we don't really need to know any of this, but what I would like you to know of is this part here. Now, let me just get the cursor there. Yeah, perfect. So we have the sensory, and often the sensory neurons, are referred as afferent, okay? And then efferent are referred to motor division of the neurons. So I often say, you know, a really good example of the how it works within the peripheral nervous system and how quickly it works. So we think, oh, this computer is fantastic and we can do Zoom calls and this computer is great and blah, blah, blah. But the speed of light, if you think of what happens with your sensory neurons, you come in, might be a cold evening, you decide, I know that that radiator is hot, but maybe it's scalding hot and I put my hand on it. Now, before there's a normal reflexive thought there to say, okay, take it off. The sensory neurons go all the way up into the brain. There is another part there called interneurons. The interneurons go, bad idea, James, send more neurons down, and James goes, boom, and takes his hand off. That's all happening for us at light speed, milliseconds as such. So when we talk about neurons, generally we're referring to the three types: the sensory neurons, which are often referred to as afferent, the motor neurons, which are referred to as efferent, and that's the interconnection between them. That's how they play their role within us. The rest of it, it's information that you can use, but it's not too important. So the neuron, okay, so you've got some pretty cool videos out there explaining the neuron quite easily. We take information in through these dendrites that you can see here, and dendrites actually is coming from the Greek word tree. So you can see that these projections look like a tree that maybe the leaves have fallen off. In the middle, we have the cell body, referred to as the soma, and then we have this thing called the axon, which is a slender projection that goes from here all the way down, and these are called synapses or terminal buttons or terminal synapses, and they send the message on to the next part. Okay, so really what happens is you get the message, oh my god, no way, and then down here when it's passed on, yeah, way, oh my god. Okay, in between that then we have on this axon these things called the myelin sheath. Okay, now disclosure, or a little disclaimer here, I'm not literally saying that the myelin sheath is a HDMI cable that you use for your TV. Okay, what I am saying is I'm using an analogy here. Your message that you want to send down the axon, so we want to send a message down here, but in order to send a good message that has high quality or high definition per se, it has to be insulated properly and adequately. If it is insulated from these myelin sheaths, then the message will be sent much more effective and much quicker than again. Okay, the neural impulse, or it's often referred to as the action potential, sends that message down, and the terminal branches are at the end then that send the message on. Okay, this is the sending neuron. This is what it looks like. This that there could be, and again, you're going to see some beautiful images now shortly about how many of them are within our brain or within our body and how they interconnect. But in a diagram to make it easy for us to understand, up on one side we've got the neuron that's sending its message down along the axon, and here is the synapses, and they're passing over to the receiving neuron. Okay. In the receiving neuron, then, we have these neurotransmitters. So the neurotransmitters are the things that you'll often hear as like hormones and things like that, that pass across there, stuff like serotonin. Some of them are excitatory and some of them are inhibitory. Okay, and what happens is there's this gap here, this synaptic gap. Okay, sometimes referred to as the synaptic cleft. If I had my hands like that, okay, now I don't know if that's a good image or you can see, there's not much of a gap in there, but the gap between the synaptic cleft and from one neuron to the next is even probably 100 million times smaller than that gap that I have there. It's tiny, there is a gap, and they have to pass over that gap. If they don't, they're taken back up here, and that's called reuptake. So the messages or the signals that are being sent down, if they're not taken across, they come back up. Some make it across onto the receptor sites. They're lodged into these little areas here. Fantastic if that happens. And then some of them are stopped from going across. And again, we'll come back to that later on when I'm giving you an example of how some medicines work in terms of SSRIs, okay? So some of them, as I said, are excitatory. So again, you think depression, anxiety, stress, all of these things. Something like depression generally has a bifold action to it. And when I say bifold, what I mean is it's generally predisposed by one of two actions. You will have people who are highly anxious and cannot rest at all. Okay, highly anxious and feel really difficult. I cannot rest. I cannot even sit still and eat. And then you have the other people who are on the flip side of it, where all they do is sleep. All they do is, sit around. They might eat, they might gorge, all of them things that are there. And our neurons are actually part of this. Our disposition when we go to get, when we go to get antidepressants are part of this then again. So if I'm the person who is struggling to get up in the morning, 9 times out of 10, I'll be the person who gets an antidepressant that is an excitatory neurotransmitter. So something like Lexapro and things like that, they tend to give people a little boost in terms of physiology and their components then at that level. The flip side of that are the ones that are inhibitory. So they're the ones that tend to be sedative by nature. You think of historically of Ireland in the 80s, mothers who are struggling with postnatal depression, they were getting things, colloquially referred to as mammy's little helpers, that what we now know as Valium. So they were a sedative by all accounts, and they would, as it says, chill neurons out. It just calms down the messaging signal, and it slows the whole system down then again. Now there's other parts to it, but essentially that's how that part of it will work. One of the key components that happens up there is this thing called acetylcholine. Again, I'm thinking the likes of Aidan and Fran and anyone else who's involved with personal training or any of that kind of gym instruction stuff, you probably have come across acetylcholine before, because in fact, any sort of muscular action will need an element or a chemical of acetylcholine. Okay, it enables muscle action, learning, and memory. So the function of what we do, it's another one of them that revs up this action. So you could probably think of, myelinated sheets, you can think of actin, you can think of how muscle fibers work. In order for that to happen, they need this starting signal, and that starting signal is something called acetylcholine. We don't have the molecule of acetyl within our system generally. What we can get from foods is stuff like is the molecule of choline, and we can build that up. So a lot of greeny things like spinach and broccoli would have high levels of choline in terms of that then again. And you'll see people who suffer with something like dementia, Alzheimer's, any of them things that are trying to aid with that. Obviously, it's a high content of omega-3 and B12, but acetylcholine plays a part within it then again. So as I said, we don't really, or a lot of the time, we haven't really understood the brain until we have dysfunction. And sadly, something like Alzheimer's, which is a neurological deterioration that can occur in middle or old age due to generalized degeneration of the brain. Okay, and that's when we start to see a slowing down of neurons. That's when we start to see myelin sheets at a microscopic level being destroyed. Okay, so we're going to look at some of the great thinkers about the brain, not in great detail, just a little quick quote in terms of DNA and how we look at DNA and chromosomes and, how they're divided as embryos and stuff like that. We look at how it started with the brain blossom worldwide is something that has taken hold in the last, I'd say 20, 25 years, definitely in the 90s. I think I said it to you before. Bill Clinton, who was in charge of presidency in the US at the time, declared that the 90s was the decade of the brain. And actually, he wasn't too far wrong. We got lots of really great things that came out of that decade, but, more so now. And then there's definitely structure of modern brain research. So again, anybody who studied anatomy and physiology, anybody who studied biology will often say, okay, two major things here. Let's look at the structure of something. What way is it made up, the brain, the different parts, the components, different levels, and then the function. So what is its role? What's its function then? Okay. And we're going to go through that then again. So here's the quote from Francis Crick. Then again, probably all are aware, the father of DNA said, "You, your joys and sorrows, your memories and your ambitions, your sense of personal identity and your free will are in fact no more than the behavior of a vast assembly of nerve cells and their associated molecules." Again, very cold, very clinical. As I said to you a couple of weeks ago, studying something like behavioral analysis for people, who want to study psychology, you go into a lecture hall and they basically make out that all of behavior can be manipulated, and that it's all just biological. Kind of undermines a lot of what we try to do on a day-to-day basis in terms of our relationships with different people. And there's lots of sub pieces of research that would definitely discredit some of the work that's there. Okay, so 3.5 billion years of life on Earth, really starting off at this bacterial level. Let's just see it's up there. And then when we look at it, there's definitely features that have occurred. So on the left, on the bottom here, we're talking about millions of years. Obviously, anyone who's got an understanding or a study of evolution in the past will probably have seen some of this, that generally what started to occur was the weight of our brain started to get really heavy. And it actually peaked up here, as you can see. But in probably the last 10,000 years, it's decreased in terms of weight. Then again, uh, we'll get into that now in a moment. It's the weight has decreased, but actually the components of what we've done have stayed the same because it's actually been compressed. And there's definitely the fissures of the brain. If you were to look at an anatomical picture of the brain, the succulus of the brain, all of that would be explained. But why is that important? Well, the heaviest part or the heaviest time that we had in terms of brain components was Neanderthal man. And it's funny because Neanderthal man, I remember we had an Irish teacher in school. I always laugh about this. Probably not a great thing or a memory at this stage, but I remember when somebody got questions wrong, he used to call you a Neanderthal. Not great for confidence, I would imagine. But you know, I often think, you know, I got my own back on it when I'm thinking all these years later, thinking actually Neanderthal man had quite a large brain, really, when we look at the evidence that's there. So in terms of how things started, probably 60,000 to 30,000 years ago, things like art started to show up, caves, cave dwellers, Homo habilis. You'll see it across different cultures, particularly Egyptian and Greek cultures would have lots of these places looked after. Language and maths then around 5,000 years ago, science 3,000 years ago then, modern computers about 70 years at this stage, cell phones, ICT at this stage today, and then understanding repairing our brain. So we actually are at a part, and if you go back to week one, I'm just wondering now, did I speak to you in week one about Jodie Miller, who had a hemispherectomy? She had half of her brain removed.
Yep.
Yeah.
Well, that was cool. It was like, yeah.
It's actually a video, I think.
Yeah, it was a video. Yeah, it was a video, and then we did a little discussion about it, I think, Seamus. But really, the neuroplasticity of the brain is a key component of when, how, and where they started to change their thinking on all of this. So it gives us a lovely backdrop. This diagram isn't great on the slide. When I send it to you, you'll be able to, zoom in on it and see. These are all the different parts of the world where this brain blossoming foundation is starting to put millions into understanding the brain and the neurophysiology of the brain. Major centers in Jerusalem, major centers in the USA, and then a big center then within the UK as well that they've just announced. Okay, so Norman Foster Center, then again, that's what it is there. You can have a look. Some people think that's absolutely horrific. If you do, you're in line with my wife. She doesn't think it looks great at all. But actually, over all of the windows, the idea is to make the building a brain. And these are basically neurons that are across it, attaching everything that's there. There's another mock-up from outside it. And this is the way it would look from inside. So essentially, they're trying to create something that looks a little bit like you're inside a brain. You can take from that what you will, whether you're a fan or you think it looks like there's just mold and some kind of moss growing outside it. Then again, really the brain and now the structure of modern brain centers work off neuroscience and neurology and cognitive disorders. They look at them cognitive disorders, like we said, Alzheimer's, dementia, anything that's associated to brain disorders, human and primate behavior and cognition. So yeah, there's still lots of research being done with, different great apes, excuse me, and certainly with bonobos, which are 99.2 identically genetically identical to us. So huge amounts being still done in terms of that. Plasticity and development of neuroscience is a growing part, but they all feed into this new factor, like I've said to you, this Leonardo da Vinci generation is what they're calling it, because it's taking the maths side, the neuro-computational side, and looking at the theory as well. So it seems cold when we just use quantitative research, but quantitative research will get us closer to a more empirical, i.e. robust, i.e. more accurate answer of what we're looking for. Okay, it's done through this theory-driven brain research. So it's partly neurobiology and medicine. It's partly psychology. Thank God they invited us to the party, says the psychologists. Computer science, applied physics and engineering. You will see why now shortly when we look at something like connectonomics. The growth of theory and modeling as well. And some people may have come across some of this stuff in the past. And if you have, fantastic. If not, it's certainly amazing on that level. So the big question is, what does it mean to understand the brain? If we were to break it down into this side of the spatial scale of the brain, you can see on the right hand side, if we were to look at simple proteins at a nanometer level or pushing up towards the micrometer level, we would then be looking at synapses. Okay, we would then be looking at neurons, as I've said to you in the past. As you come up to the millimeter level, then it's them circuits of how they interconnect together. So if you think about them circuits that connect together, we've already spoke about that big classification or differentiation tonight: central nervous system, peripheral nervous system working together. Okay, again, I often think back to my mom, really great explanation of central nervous system, peripheral nervous system. There was a phrase when I was growing up, I don't know whether people still use it, but there was a car accident with a lady who used to live down the road. And I remember everybody used to say, oh, she broke her back. That was what they were saying, she in a wheelchair, she broke her back. And actually, she still had feelings within her legs, but actually had no use of them. So it was actually that a component part down the lower part, near, I suppose it would be probably a little bit lower than the lumbar. That was the bone, that was the area that got broke, inverted commas, and she could no longer walk then because of it. So my understanding of the central nervous system was it's the brain sending the message down the spinal cord, and if the back, the spinal cord is broke, then that actually means that we'll cease communication to the rest, or it could cease information going to the rest. Now, information here is still had use of her arms, but actually because of the part of the spine that was damaged, no use of her legs, but could feel them, so had sensation in her legs. Again, these are the levels that we're working on. So the centimeters, then again, brain regions, the whole brain, as you can see on the right hand side. I'll just put the cursor over it. And then when we work in meters, we're referring to the body then again. Okay, so recent brain excitements, again, that's excitements. We'll look a little bit more in detail at 1, 2, and 3, optogenetics, light activated brain circuits. There's one little clip in it that I might show you, and then computer simulation of the brain, again, Blue Brain Project. Massive amount of money put in it worldwide then again. So modern neuroanatomy is all about mapping and visualizing the wiring diagram of the brain. So where does neurons activity start? How does it start? And what are the localized functions of certain things? Okay. Now, I would be remiss, I would be a really bad lecturer if I didn't say that even with loads and lots of localized areas, like I say to you, is the hippocampus. It's really like a memory center for us, particularly our long-term memory. Even for the hippocampus to work, you're talking about, millions of neurons firing off for me to have a memory. So I gave you the memory there of how I learned the central nervous system, for example. Millions of different neurons are firing away. Okay, so we have really two big figures within it. Okay, Camillo Golgi, you probably are aware of the Golgi tendon organ. You might have heard of it. If you haven't, it's basically a reflexive organ. If I was to stretch my leg back, and you think about sports, if I was to reflect, there's that reflex, and it's basically based off this guy's work here, Golgi. The other one then, Raman Kahal. Again, these were people who got Nobel Prizes. Golgi believed that they were all one unit, but now we understand that they're not. They're individualized units, like Kahal said, that link up. So they link up as they start to pass messages on. Golgi, of course, for the time, you cannot criticize them, you cannot give out to suggest that they were all just one unit working together, because when we think about all of the things that happen within our body, they are all working, but just not off the one system. Okay. Again, is there anything there? No. So there we go. So it just explains what I've just said there then again. Okay. So I'm just wondering now, I think this video could be a little bit longer. I think it's a 10-minute video. I'm going to send you on that link down for Connectomics. Essentially what it does is it takes the brain and it slices it. Okay, so it's unbelievable, this process. If you can get a chance to watch this video at a later time, I think it's quite a long one, but it slices the brain, okay, into, I mean, really thin slices. So something like, I think they say in the video, like 70 million or 70,000 times thinner than one of the hairs that's on your hand or your arm. Okay, so you can imagine what it does. So what it does is it slices apart the brain, and then it puts it back together in terms of a machine to try and loosely find out some of the actions that occur within it. Okay, so here's one that they slit apart and then they put back together. They added dye in here. So you'll hear, I'm going to show you a thing now in a moment called a brainbow. So like a rainbow, but it's a brain. Okay, this is essentially when dye is input into the brain for us to understand and try to localize the function of different neurons here again. So which is the kind of image that you're going to see here now. Okay, let me just go back there. Let's try play this. This is a tiny part. So even within that part, look at the multi-modal amounts of things that are in there and the different functions that they all undertake. The ones that are more towards the pinky red color, they tend to be the ones that are sustaining blood. So there's blood vessels that travel through then as well. And you'll see it goes right into the dura mater then again. So really all that the video is trying to show us is that there is a multitude of different things that are even within a small component of this connectomics when they're trying to find out and localize function of what's happening within our brains. Obviously, this has been done and performed with a mammalian, so a rat as such. But you know, obviously they're trying to find. So what are some of the things that come from it? Well, for the first time, we'll have a real blueprint. So not an image, not a kind of a scan. We'll have a blueprint, the anatomical foundation of a whole healthy and sick brain. And really, as I said to you before, a lot of why they're doing this is to try and localize some of these elements that cause dysfunction to the brain. If there's dysfunction to certain parts, can we then input electrodes into the brain to fix certain parts or to help other people live sustainable lives? The answer is yes, of course it is. So we may start to bridge the structure to function. So remember what I said to you is any organ that you study, again, people who have got gym instruction, personal training, your understanding of anatomy and physiology will be aware that we've the structure of something, how is it made up, and then the function of things is really important. But there's always been that big gap between why. So with this new research, they're getting close to it then again. So this is a brain image that you can see there with dye input to it. And these became really famous. There is lots of different pieces of art in different museums around the world, but this is called a brainbow. Okay, so these are the neurological pathways that are there. The synapses, you can generally see them by their kind of pulpy kind of ends. Then you can see these elements here that look like dendrites or trees, as I said to you at the start. I think Greek word for it, yeah, coming from oa. So again, that's the image that's up there. When people often say, why did they do it then again? Well, it allows them to then function in here. So this is probably a really diverse image. Now, an image that I don't understand, if you do, fantastic. The reason why it's important here is essentially what's happening with these red sections here is if them neurons are the ones that are causing dysfunction within a brain, they can then completely hone in and accurately try to either burn them out, okay, so through electrodes that they have, microscopic level, to try and see if that will work, or can they add something in that will help? So sometimes what happens is the electrical impulse, the spike, the action potential won't fire away, and it's like me trying to pass a message on. So I pass it to Mick, and Mick tries to pass it on then to Donald, and Donald tries to pass it on to Emma. But if Emma can't pass it on then to Francis, then it's not going to get to Linda, and so on and so forth. So therefore, this is the same kind of prospect here. They put that dye in there, the discoloration of the dye, then again, to try and find out where it is the dysfunction is occurring at that level. That's the level of detail that they're at with this genetic staining of neurons in vivo there. So the light microscope, micrometer resolution, then again, really fascinating stuff then. In relation to this brainbow and transgens then again. Okay, so here's just some more images there of the neocortex. And what we have as well is the hippocampus, which is up here. This is the one that we spoke about, the hippocampus. That's our brain, sorry, the hippocampus, it's our memory center. It stores and houses some of our memories that is there. As you hone in on different parts, then again, you can see the different colors and outlines of these motor axon neurons, which is really detailed. Sorry, I'm just going to close this. So with them, again, then what we have is these motor axon neurons, or we have sensory neurons, and we also have interneurons. But these images are microscopic, and they're really important. So what does it do? What is the prospects behind it? Why is it important? The structural basis for learning in the brain. So it gives us really good detail for the first time. Tagging and genetic characterization of the different cell types. So building blocks in a given system. The retina of the eye, for example. So how can they be replaced? How can they be repaired? Same thing again, or tracing short and long range connections in the brain circuits. This is a big part in terms of people who have any sort of amputee would have this type of stuff. And we're going to look at this connection between our brain and machines now in a moment, the BMI as it's referred to then again. So the brain machine interface, or you might say it as BMC, sometimes brain machine computer, but it's generally referred to as a BMI. So you might be thinking, oh, body mass index, but actually when we refer to it in neuroscience, online BMI requires reading the electrical activity, its electrical language, its code in well-defined functional regions. So again, this might be a lot for us to be taken in, and it probably is. Remember, we've the recording of this as well. But the reason why it's important is it's allowing us to take a message from the brain and input it into a machine directly. So actually what I'm doing here, message from the brain out to the air through the mic and into a machine. But this is a different type now. So we've got the brain. We may just have this function with a neurological pathway. We hone in on it and we see these spikes that I've referred to as earlier. The spikes are the action potentials that are really strong signals. We need them. If they're there, I can pass the message to Francis, and so on and so on. We can keep passing them messages on. If that doesn't happen, what can we do to try and help with these situations? Or if somebody has some underlining issue, what is the thing that we can do? Well, you may have seen this before, and if you have, again, fantastic. If not, no real concern or worry. This is the brain activated robot arm. So this brain activated robot arm is allowing for really us to send a message from the brain down to the spinal cord and then in through our arms, which allows us to create pincer movements that we have there then again. But it also is allowing us to take a message from the brain into an artificial neural network, and us to be able to grasp things or use these robotic arms then again. I will just say the video that I'm just about to show you is with a rhesus monkey. I can also tell you that these monkeys are 150% looked after. The researcher is very outspoken about ethics, Andrew Schwartz. Again, really outspoken about ethics and consideration. So although the monkey has a brace around its neck, this rhesus monkey, there is in no way it's being harmed. There's actually live feeds into the laboratory where Andrew works and stuff. So just the people are a little bit concerned about any of that, but this is what we see. This is what we can see with this, where there's been dysfunction and they're using the artificial neural network here. Okay, so I'm just going to play it. I'm not going to leave it play for too long, but you'll get the image of it. You get the idea of it pretty quickly then again. Okay, so there you go. They just putting the food on this element here, and then again, this is all through the neural network. I'm pointing at the screen there as if you're in the room with me. This is all through the neural activity network. Okay, it's an artificial one. So essentially, this little rhesus monkey is feeding him or herself. Not sure if it's a male or female, but they're feeding themselves. So the movement of the robotic arm is all being done through that artificial neural network. And as I said, anybody who, like me, loves animals, I can tell you now that there's a huge amount of ethical guidelines and boundaries that are in place there then again. So what we've done here is we've went from brain to arm. Okay, so we've gone from brain to arm in this case, and now we're looking from machine to brain then. Okay, so again, this is something that you'll probably be aware of. Our basal ganglia, do you need to know it? Absolutely not. All we know is that highly important part of our brain in terms of amalgamation of different functions that we need to perform. So this will be a normal brainwave activity on an EEG, so electroencephalograph. So it goes and stops. Okay, that's a normal one. In Parkinson's patients, then you can see that it's very much stop-start, and even in an anecdotal kind of way, you would think of why that would impact somebody with Parkinson's disease then again. So tremors would be definitely one of them things that's there then again. Okay, so in terms of taking actions from, we've gone from brain to machine, we're going from machine to brain this time. So you may have heard of these aren't as niche at the moment. There's pulse generators put on just in front of the heart or placed on the heart, and then through stimulating electrodes, it goes down from the top of our brain into the basal ganglia, and it allows us to perform action. Now, sadly, I just checked this earlier. Yeah, now it won't play the after, but this is a patient prior to it. It's not really a good example, but you can see that they're kind of walking step by step, very labored, difficult to do. Then again, really sad, first of all, and pincer movement seems to be limited, difficult to do. Even shaking hands seems to be, or shaking of the hands seems to be difficult, and you can see the tremors then even in the feet. The other video essentially is just showing somebody walking fluidly, somebody clicking their fingers, somebody clicking a pen, and essentially the patient, male, 58 years old, and this was when this was input. So the being able to amalgamate them together then again shows that this deep brain stimulation works for people, particularly with something like Parkinson's disease then again. So massive strides that have taken place over the last 15-20 years then again. So their future challenges basically is that although we've seen the rhesus monkey there moving the mechanical / robotic arm, one of the main problems that's going to happen is essentially they're trying to improve closing the loop. And closing the loop is basically referring to people want to feel things. So I just probably see me drinking from this evening. When I pick that up, I can feel that it's cold. I've deliberately made it cold. So these interfaces, they're trying to get closer to real simulation, excuse me, of what's happening there again. They've got a certain capacity. They can take it from this artificial neural network, but in terms of creating sensory processes on the thing that's lifting, it's still not there. So even with all the computational advancements, it still hasn't got there. Okay. Here is an image then. This is an unbelievable image. Again, for me, it's an unbelievable image. This is the electrical activity within the brain. It's 10,000 neurons and it's color coded that way. This is the kind of activity that takes place solely even when somebody is beginning to talk. So this is all taking place at a neurological level. And it's really important. If you go to microscopic level, this is what it looks like. Again, it's an optical imaging of the cortical circuit. So again, in between that cerebral cortex and the basal ganglia, then again, it just looks like, you know, tadpoles moving, etc. But it's actually a really amazing image as well, where you can see all them pathways connecting as they go then again.